Good evening, everyone. Thank you so much for joining today's People in Clinical Trials webinar. My name is Victor Ramezi, and I am your host. Today, I have Dr. Pindile Kumalo, who will be giving us a talk on what constitute a quality clinical trial, time as a fundamental factor to quality clinical trial. So what we're going to do is Dr. Pindile will take us through her presentation, and at the end, we'll have the opportunity to ask questions and also give any comments. I would like to encourage each and every one of you to be as interactive, and let's make this very beneficial for everyone that's attending. I am going to be recording this session, and the recording will be made available via YouTube, and I will share the link to the YouTube video on my LinkedIn profile. So briefly, to introduce Dr. Pindile Kumalo is currently an internal monitor at Vets Reproductive Health and HIV Institution. She is a homeopathic medicine practitioner by profession, and she's currently studying towards a master's in public health at the University of Western Cape. That's briefly about her. And without wasting any more time, I would like to hand over to Dr. Pindile to maybe say something before she starts with her presentations. Dr. Pindile?
Good evening, everyone. Victor has already introduced myself. I am Dr. Pindile Kumalo. I'll be the speaker for today. Actually, just for you guys to match the voice to the face, I'll just show my face just a little while, then I'm going to continue my presentation. So, hello, everyone. Yeah, this is me. So, yeah. Okay, I'm ready for the presentation. I'm going to take you through this few slides. So, today's my topic is on what constitutes a quality clinical trial and time as a fundamental factor to quality clinical trial. So it's more likely that we are doing a recipe to produce a quality clinical trial. I'll just make a silly example that when you are baking, you can have all the required ingredients. However, the product might not be what is supposed to be like. So today I'm just going to be taking you through the ingredients and how to use them in order to yield the required product, of which our product in this instance will be the quality clinical trial. Okay, so let me start. So this is it. Okay. So this is just a disclaimer that the content of this presentation is just based on my experience and also my views and my observations through my career in clinical trials. Okay, so these are the constituents of a quality clinical trial as per my observation. I've listed them and they are not in order of importance. I am having training, protocol, and obviously there's no way you can have a clinical trial without a protocol. We have SOPs, quality management system. I have data and there's a question mark there. And there's also time, which is also forming part of the bigger part of today's presentation. So I'm going to be taking you through each and every component of this aspects. So what about training? So we know that GCP, which is the good clinical practice, it is the prerequisite for everybody who's part of a clinical trial. So it is important, we all know that it is important for everybody who's into clinical trial to be GCP certified. Okay, so this is just one of this is the prerequisite, as I've said already. Also, as part of training, I feel like training is a very, very, very fundamental ingredient into producing quality clinical trial. I'm not gonna count GCP as part of the training because without GCP, there's no way you're doing clinical trial. There's also trainings that are important. For example, we have the ABC of quality control and quality assurance. This one usually is done by the staff that are into quality, and not only them, but some of these trainings are very much important in order to harness the skills of the people that are in clinical trials. And we know that, for example, there's NEMA training and also IATA training. This training are specific to the duties of some of the people that are part of the clinical trial. For example, nurses are supposed to do like your NEMA training, those who are into HIV clinical trials, and also your IATA training is very much important for like your nurses and whoever's handling samples. So there's different parts of trainings, right? Of which these trainings, in most cases, like this one, the first three, the first, sorry, not the first three, my bad, I'm a bit stuttering. This one, and that one, they are most likely specific to different roles. How, is somebody saying something?
No, you can continue. It was a mistake from someone.
Okay, thank you. So we're having this training that are specific to certain roles. However, in order for us to have a good clinical trial, it is important for us not only to depend on the training that we get from the external sources. However, we also have things that are called like your preceptorship. This one here, it's something that is usually done within a site, like it's done internally, whereby a site will have like structured trainings for each staff in order to make sure their skills are harnessed at all times. They know how to do what they're supposed to be doing, and do the staff really understands what they're supposed to be doing, because we cannot only depend on only the required trainings, but then even if with depending on the required training, time and again, we're also supposed to be having refresher trainings, because it happens that we forget how to do certain things. So it's one of those important things, that training is very much important, as it has always been said that knowledgeable staff or trained staff is always, knowledgeable staff always has the ability to produce quality work. Therefore, it is important for such to invest in the training of their personnel. So that's ingredient number one. We move to the second ingredient, which is the protocol. But as I've already mentioned, that there's no clinical trial without the protocol. But what do I mean? Or why did I add the protocol as part of the ingredients? Because I've noticed that at times, because we know that in most cases, when we're conducting clinical trials, at all times, we have to adhere to the protocol. And we know that if we do not adhere to the protocols, it is when we'll be having a number of errors. We're going to be having protocol deviations. Therefore, it is important for us to adhere to the protocol, most likely 100% of the time, of which that is something that is not always happening. We all know that. And also, I've also put another component here, which is protocol comprehension. What is this? What do I mean by the protocol comprehension? Here, it happens that I will read a certain portion of the protocol and finding that I'm understanding it in a certain way, interpreting it in a certain way, and then start doing procedures the way I understand the protocol, only to find that I'm not doing it correctly. Therefore, in this instance, it is very much important that when you do not comprehend a certain aspect of the protocol, that you always get a second opinion from the team member or from your CRA, or most likely, or most preferably, the protocol team. For instance, not so long ago, I was reading a protocol for another study, and there's a portion whereby they were talking about the post-vaccination bloods. And in my mind, what came to mind was that, oh, that means these are the bloods that have to be drawn after the participant have been vaccinated, only to find out that I was incorrect. Just imagine if I already interpreted that and put that into practice, there was going to be a lot of PDs based on my interpretation. Therefore, it is very much important that if we do not fully comprehend, because at times it might happen that you think we are comprehending something, we all know that, that's where most PDs are coming from, but then this is the aspect of the protocol that we need to adhere to, that making sure that we understand the protocol the way the protocol team designed it, and that we adhere to it at all times. So this is another factor. All right, and now we're moving on to the third one, which is the SOPs, the standard operating procedures. But then what about the standard operating procedures? I've read a number of articles on quality. It's been said that a good standard operating procedure is being designed by all the members that are involved in that particular duty. Therefore, it is important that if SOPs are being designed, people who are part of that SOP is being designed for, they have a say, because they are the ones who are doing the duties day in and day out. Okay, because we have like the SOP management cycle, whereby we'll first have to go through the SOP needs assessment, that, oh, okay, maybe there's been this and that and that. Okay, what do we need to do now? We come here to the SOP development, but then during this process or during this stage, that's where all the people that are involved need to put in their views, and then it will be implemented, obviously, and then it will be evaluated. But then also, as I've mentioned, the time factor, it is very much important here that how often, because often is time, isn't it? So how often do we evaluate these SOPs? You know, because things have changed. For example, with COVID, I know that most sites, maybe they've changed a number of their SOPs because of COVID, because now we have to incorporate COVID in some of the things that we are doing. So it is very much important that SOPs are being evaluated. Okay, in most cases, they're being evaluated maybe once a year, or maybe it can be twice a year, or if there's a need. And again, the importance of having SOPs, what it does, it enhances smooth flow of work. Because if you have a clearly identified or a clearly outlined SOP, it always makes things easy for the staff that are working on a particular project. Even if you can take somebody who hardly works on that particular project, but they are delegated for that particular study, if the SOP is clearly outlined, there's not going to be a number of errors. As you can see that I've mentioned that having a strong SOP will reduce errors. It also improves and sharpens the skills of the workers and also their performance. And if their performance is being enhanced, guess what? We are going to yield a good quality work, isn't? And also it eliminates the guesswork. Like, oh, do I have to take 2 meals or do I have to take 3? Hey, it eliminates guesswork. You know that if you have the SOP and the protocol by you, chances of making mistakes are usually less. Therefore, those limit the number of errors at all times. As again, if we're talking about quality in general, if we say something is of good quality, in general, we're saying that something it has no errors. But we know that in clinical research, there's no way we can conduct a trial without errors. But the main aim of conducting quality measures is to reduce the number of errors. So in order for us to have clinical trials that are of minor errors, we must have these ingredients that I'm taking you guys through today. Okay, so this is the importance of our SOPs, making things flow easily. People know what they expected to do and they know what they're doing. And it also improves and sharpens their skills, right? The next one will be the quality management system. Again, before I go through or I delve into this one, it is very much important for everybody who's involved in a clinical trial to know that to maintain quality is not only the quality personnel's responsibility. However, it is everybody's responsibility to adhere to all the quality measures that are put in place. Okay, as much as the quality personnel are playing a big role into maintaining quality, but it is important to know that it is everybody's business. Quality in clinical research is everybody's business. However, when it comes to this one here, the quality personnel, it is their main role. Okay, so let's go through this one. Again, if we are having these as our ingredient, it is very much important that we have a clearly defined clinical quality management plan. As the clinical quality management plan will outline, it has to clearly outline the quality assurance and the quality control activities for that particular site. And again, I will just go through the QA and the QC activities. For instance, we can have QA and QC activities, but then what do they entail? We must make sure that they are checklists that are used. And this checklist, in most cases, for example, if you're packing a bag, you're going somewhere, you know what a checklist is. You know that you must carry your toiletry bag, your this, your this, your that. So these checklists are important in reminding us that, oops, I have this checklist. It has all the things that I'm required to pack. However, I've packed already, the bag is sealed, and I'm already on the way. But then if you go through the checklist, that's where you realize that, there's something missing. So it is important that as part of our QA and QC activities, we're having checklists that outlines all the required things for each and every aspect of the clinical trial, because it helps us to see how far are we with the process. Are we doing it right? Do we have everything that we need? Did we miss anything? Did we add something erroneously? So it's very much important to do that. Also, we must have what? We must have tracking logs, right? In terms of tracking logs, we also need to track that. How far are we now? They said we must enroll 5 people, for example. How far are we? We must track. We must track. Also, with these tracking logs, we're not only tracking how far are we, but we're also tracking the number of errors that are being done in the site. But then the most important the most important thing is that once we have tracked, what do we do? What do we do once we have tracked and see that they are trends? Therefore, that will bring us to the CAPA plan, the corrective action, and also the preventative plan. It is very much important for us to go through this thing. Tracking logs, there must be in check at all times. And also with these tracking logs, it also has to do with, for example, they're asking, guys, how many loss to follow-ups do we have? We're gonna be scratching heads now. We do not know, because we do not have logs that are tracking all the events, or the events of the clinical trial. So all of these things, they speak to quality, because we need to know, as we are doing this clinical trial, how many people are lost to follow up? How many people have withdrawn consent? How many people we've enrolled? How many, it's very much important for us to have all of those tools at all times. Should there be mistakes, what do we do? It's very much important as well. I'm gonna go through the CAPA plan later on in terms of the timing factor, and also the quality measures. Also, I've already made, with the quality measures, do we have tools that are being used in order for us to evaluate quality? And also, once we have evaluated this quality, what do we do? How often or when do we disseminate the results or the findings? Because sometimes you will find that we get accustomed to doing things the way we do them, and in such a way that we hardly see when we are now starting to make mistakes. Therefore, or sometimes you'll be doing QC or QA of the file, you find a query like, no, this is just a minor query. I would advise people that even if you think it's something minor, it may be minor to you, but not minor to other people. Because sometimes, from my experience, sometimes it happens that we're like, this is something minor, and then the external monitors will come in, and guess what? Those things may become part of the findings, and you're like, I saw it, but I didn't tell them. So we do not need such things. So take each and every query as serious as possible, no matter how much you think it is minor. But remember that we are doing all of these things in order to harness a good quality. It is not your study. It is not about you as a QC or QA personnel, but make sure that you take each finding as serious as possible, and make sure that these findings are disseminated timelessly, so that people who are doing these things, they know, this is what I'm doing. This is what I'm doing wrong. Because sometimes if I'm not told that there's something I'm doing wrong, I'm going to continuously do it. And it's going to take us a lot of time to go back and fix those mistakes. So it is very much important that if we have findings, let people know on time. Let people know on time, so that we'll then have a way to resolve those queries timelessly. Not that, because sometimes in other sites, come and again, you'll find that if there's monitoring visits, people will be pacing up and down, trying to fix query, trying to fix this, to fix that. I think it is very much important for us to always be audit ready. Do your work as if there's an auditor coming the following day. Do your work as in there's an auditor coming the following day, because when the auditors are there, you're not going to be pacing up and down, because you'll be knowing that you've done all you could. you know? So it is very much important for us to have all of these quality tools in place and use them correctly, so to say, because we can have all of these things in place. For instance, I haven't listed everything. This is just an example. We can have all of these things in place, but then find that we're not using them the way we're supposed to be using them. Okay, so it is very much important for us to do things like correctly. And another thing, I believe that here in this webinar today, we are having different personnel, not only quality people. Am I right? Okay, if yes or no, it's fine. But then it is very much important that every query is taken serious by either the investigators, by the nurses, and by the pharmacist. Okay, right. And then the next part that is forming part of the quality management system will be the data management reports. These reports are also a way of harnessing good quality. Also, it is very much important for us to attend to this report also, Nakona, timelessly. It's a matter of like time, because if we do not attend to these things on time, excuse me, if you do not attend to these things on time, it then kind of not jeopardizes the work that you are doing, but it also affects data. Also, as part of this quality management system, I just want to add something that sometimes we take certain things light to a point that we forget that in clinical trial, it's more like a cycle. Yes, we are part of the circle whereby we're collecting the data and all of this and that. Remember that if we are not collecting the data that we're supposed to be collecting, that definitely sure it will affect the validity of the study that is being conducted. And remember, that can also affect the statistical analysis of the data that we are collecting. We must always remember that it doesn't end with us in the clinics whereby we are collecting this data, but it goes further and further. Because remember, in terms of like the statistics and all, whatever trial or whatever study is being conducted, there should be like the reliability of that study and also a validity of that study at all times. Must remember that as much as we may not be statisticians, but there's more after we are done collecting the data. And also when it comes to monitoring visits, this is another tool that is used as part of the quality management system, because these are the tools that are helping us to know whether what is it that we are doing correctly and what is it that we are not doing correctly. And it also shows us, for example, you can, as I mentioned earlier about the protocol comprehension, that it happens sometimes that you misinterpret certain things. For example, during the source documentation development, if most of the things are misinterpreted, chances are you're gonna do a lot of mistakes. Therefore, with the monitoring reports, it can guide us that, guys, site so-and-so, you have to change your source doc. This should be put like this, because remember, all of these things, they are there to harness good data that will be used for whatever study that we are doing. So that's that. I think I've just spent a lot of time in this, but this one is the bigger component, or it's the bigger recipe. If we said with the other ingredients, we are pouring like 5 mils here, we are pouring 20 mils, because this is the bigger part whereby quality is being done. Right. And then, as I said earlier, that this is more like a recipe. So we've already gone through the ingredients. As I said earlier, that these are not the only ingredients, but for me, the ones that I've listed are the most appealing to me. Okay, so we have all of these ingredients. We have seen that, oh, okay, we've already learned on the previous slide that how we should use each ingredient. But then how are we going to know that whatever we are doing is correct? Therefore, they are key indicators. Okay, these key indicators that I've listed here, they are adapted from the dates policy of 2019. So these are the key indicators. We're having informed consent form and informed consent process. If the informed consent process is not going accordingly, that might be showing with guys, there might be something wrong with how we are doing this thing. There might be something wrong. Also, in terms of the assessment of understanding of ICF, is very much applicable, isn't it? So we have to make sure that, are we really entering participants that are fully understanding the study that we are conducting. Also, with the eligibility criterion process, are we really enrolling participants that are really meeting all the inclusion criteria, and those and they are also like not meeting the exclusion criteria? Because sometimes it happens that we find ourselves enrolling people that are not eligible for the study, and that is a big no-no. So this is an indicator. These are the things that indicates the quality of the work that we are doing. Because even when we are doing QC and QA or internal monitoring within sites, these are the documents that we go through, looking for quality, looking for whether was the protocol required tests and procedures, were they done accordingly? Were they done on the required time? If they said you must draw bloods at week 24, were the bloods really drawn at week 24? And all of those things, this is where we address. So it's very much important to look at these things, the indicators, because it shows that are we moving towards the right direction? And also at the same time, these indicators will tell us whether they will tell us whether we are doing it correctly or we're not doing it correctly. So basically, if you look at from number 1 to number 11, these are all the things that are being monitored in most cases. So you can go through them at your leisure. It's there online. So yeah, it's just one of those things. Yeah, so I'm not gonna go through all of them because I'm just having limited time. So yeah, so just going through all of these indicators, also in terms of in terms of visits and also the missed visits. Because if a trial is having a number of missed visits, guys, there's something wrong going on there. Why are these participants missing their visits? You know, so those are the things that we need to go through. Because also, again, in terms of this, in terms of the visits, sometimes you may find that, for example, let me just make a quick scenario. Here's an ICF process. It's been done maybe not correctly. The participant was not really comprehending what they are putting themselves into. And then later on, we find that they are eligible. Obviously, we put them in the study. Everything is done according. But then later on, they realize, man, you know what? I mean, I don't understand. I didn't understand that I have to be like giving my blood time and again. And then you find that now we're having people that are withdrawing consent, that are doing this, that are doing that, you know. So it is very much important that from the beginning, from the ICF process, that people, the participants are explained all the processes accordingly so that they know exactly what is expected of them during the conduct of the clinical trial. So yeah, that is that. And then the second part to my today's presentation is the time aspect of quality. Yes, we've gone through all of these indicators, right? But there's a time aspect that is very, very, very much important. I feel like as much as it may not be listed anywhere, that time is an important aspect to quality. But from my observation and from my experience, I've realized that time is very much important in adhering or in harnessing a good clinical trial. For example, these are just like different scenarios that I've posted here. For example, conducting quality checks and give reports timelessly, that is time. Because remember, if we're giving a quality check, the quality reports on time, that means that the errors will be corrected on time. And then in terms of data, we're going to be able to meet the data timelines, isn't it? So it is very much, because remember, if we're entering data a long time ago, it was required a long time ago. So if you put it later on, will it be of help? Yes, maybe it will be of help later on. However, from the site, it might not present well from the site's aspect that how every time these people are capturing their data later on. So time is a very important factor. Also, with conducting visits as stipulated, this again is the time factor. As in, like, if we said we're having 5 visits in this particular clinical trial, are these 5 visits conducted at the time points they're supposed to be conducted? Because if they're not conducted at the time they're supposed to be conducted, that can also affect the data analysis, because we're not looking just within site, but also ahead to the data analysis part, which is later happening on, of which we're not part of it, but we must also bear that in mind that, oh, for example, if we know that maybe they want to do a study and then they injecting, not injecting per se, but they are withdrawing bloods at 6 months, and then there's a participant coming like maybe 3 months later, for example, and that will be at 9 months now. Will that data be, sorry, will that be, that would not be sufficient, so to say, or it wouldn't be that much of help, because we know that there's a certain reaction maybe that the protocol team will want to see at that particular week whereby they're drawing the blood, the certain things that want to see at that time point. So if we're not adhering to the time points, that can also affect our data. Right, and also reporting SAEs at the required times, because you see, there's always that time factor. And also, here's another one, also the time participant spends on site. If participants are spending long hours on site, not attended to, for instance, that can also affect our retention. You know, that can also affect retention. And in turn, it may affect the quality of the work that we are doing in terms, you know, it will affect the quality of the work that we are doing, because we also know that word of mouth is one strong thing that happens. Like, oh, you are part of that study, you're gonna have problems when I'm spending time, like a long time there. So it's very much important for us to keep time at all times. And also affording our participants adequate time to comprehend the ICF, for instance, as the scenario that I made earlier. It also harness quality, because we'll know that we are having people in our clinical trial that fully understand what is expected of them. Because if they are not given enough time to understand, we enroll them later on, we're going to be having what a number of consent withdrawal, loss to follow up. People are no longer now picking their phones when we are calling them, you know. So there's quite a number of things in terms of the time factor. Also, in terms of the calibration of equipment, there's also a time factor to this. And how often are the apparatus or the tools that we are using are calibrated? Or sometimes, because others will notice when everybody is gaining like a 5 kg weight after a week or after like 2 days, that, oh, flip, why are all these participants having like a 5 kg gain in 2 days or whatever the case is? That is because of what? Not calibrating the equipment on time. And that is also affecting the data that we are collecting. And therefore we have to go back and correct all the issues. So therefore also the SOP reviews, I've mentioned that as well, that all this, the SOPs also need to be reviewed, not timelessly per se, maybe once a year or as per the need. So this is just the time factor that I wanted to bring. There's quite a lot of things that may go wrong if we do not do certain things during the conduct of the clinical trial on time, right? Because for example, what you call this thing, a participant can come on site, do the visit, but then if the file is not brought on time to the QC or QA personnel, guess what? There'll be the very same data that was collected for that particular date might be entered late as well. So we have to keep time. Also, there was an error that was picked up, but then the study staff was not told on time to fix it. They're gonna keep on doing it time and again until it becomes a big problem. Therefore, we have to do everything on time, inform people on time, communicate on time. If we're having queries, fix queries on time. You know, as soon everything here needs to be done as soon as we can in order to eliminate query, because at the end of the day, quality is something that is limited, or it has no queries, because the main aim for us is to have what? No queries at all. So I think just came to almost the end of my presentation. Oh yes, so any comments or questions for me? Thank you.
Okay, thank you so much, Dr. Pindile, for the great presentation. And I think most importantly, highlighting and outlining a very crucial step in ensuring that our data that is collected at these clinical trial sites can be used for proper evaluation of the medicines that we are testing. And also if efficacious and safe, they can be used, the data can be used to support registrations of these drugs, which are often life-saving. So right now we are going to open the floor for questions and comments. Please, let's be interactive. Let's ask questions. Let's make comments. It can help somebody that is just listening or somebody that is interested in the field. So just feel free to unmute yourself, or maybe because there's a large group, if you can raise your hand and then I will call out your name and then you can unmute yourself and then you can give us your piece of mind. One question from me, Dr. Pindile, as we wait for other questions. If you are shy, please use the chat function and then we can read it out from there. You know, at one of your earlier slides, you mentioned that it was talking about protocol adherence and protocol comprehension. And that is very interesting because when you're at site level, when you do training, you understand differently, and then you are the ones that are doing the actual procedures. How do we ensure that we understand the clinical trial protocol as it was meant by those that wrote the protocol?
Thank you for your question, Vic. As I alluded earlier, that at all times, if you are unsure, it is always preferable, as much as you might ask the team within site, but the most preferable thing to do to make sure that you understand correctly is to ask the protocol team, because they are the ones who know exactly what did they mean on a certain aspect of the protocol. I think it's always safer doing that, or also asking the CRAs if they are within close limits to you guys. So yeah.
Okay. Richie, you can unmute yourself and ask your question.
Thank you for this opportunity. First, I'd like to say thank you to Dr. Pindile. It was really a great presentation. You really summarized pretty much in terms of quality system, what happens at the site. So I just had I have a question, and understand that quality, or say poor quality often results from either a bad system in place or it has a human factor. But focusing on the human factor, like human error, I wonder, how do you deal with conflict? Say, for instance, you could be raising a query that you feel is relevant to data quality, but the study coordinator, or be it an investigator, tend to disagree with you. At that time, how do you resolve such conflict?
That is a very interesting question.
So we're going to shower. You'll shower, then you go to grandma.
Okay, Richie, I'm going to answer you just now. Okay, Richie, that is a very interesting question. And to be honest with you, that is something that I've also experienced in my career, that you'll be having a query, but somebody feels like, this is not I cannot fix this query because they're not understanding. You know, at times, or what we've done from where I'm from, we usually have the clinical team meetings, whereby the quality control and quality assuring team will be having a number of queries. We're going to be presenting the trends to the clinical team, to our investigators, our nurses, and our doctors, showing them, okay, guys, because the thing is, if you are approaching it in a single manner, you're going to that particular person, maybe all the time, they give you that problem. How we usually dealt with, or how we've dealt with it, we'll have like a meeting with all the clinical staff, and then we're going to be presenting all of those queries that, okay, sometimes we do the name shaming just in order to harness good quality, because other people are repeating the same queries time and again. So we say, we're having these queries, then let us break it down. How do you guys understand it? Or how do you guys tackle such? Because it might happen that it might not be the very same one person who doesn't want to fix that query. But if you put it on the table to the entire team, it becomes much more easier to resolve rather than dealing with one person, because that person might be having a strong stance that, no, I do not want to fix it because this is how I understand it. So it's better for you guys, all of you, you sit down. How do you understand this? The comprehension part goes back again. How do you understand this? Therefore, if this is your understanding, how do you think we should be answering it? So that if we're discussing it in a full manner, everybody will now, you're now going to be seeing less discrepancies, whereby all the clinical team are answering to that same query the correct way or the same way to your satisfaction, maybe. So I don't know whether I'm helping.
Yes, it's pretty, you nailed it. So thank you for that.
Thank you, Richie, for your question. Caroline, please unmute yourself and go ahead.
Hi, everyone. Thank you, Pindile. Very informative. I just wanted to add on to what Pindile said after Richie's question. So, you know, from experience, I've also learned that if there's a query, you know, it's the atmosphere that helps people to understand how to sort it out. Because, you know, if it's more of a you know, kind of pointing finger, it becomes an issue. But if it's more of, listen, we've got to get it right, we're in it together, then it becomes easier to you know, to help the other person understand things from the, you know, quality control you know, personnel. So you know, I just thought I should just, you know, put it out there as well, because it's usually the environment then that assists the other person that sees things differently to see it from the perspective of suppose, and I guess what the protocol team sees it to be. Yeah.
Thanks, Caroline. Any further comments or questions? All right. Since there are no further comments or questions, I would like to end the webinar. So firstly, thank you, Dr. Pindile, for your great presentation, taking your time to put together this presentation. It was very informative, it was very insightful, and a lot of comments are coming in that they found this presentation to be very informative and helpful. I would also like to acknowledge African Farmer Review, who sponsored this webinar. You can follow them on LinkedIn. It's a great platform that shares pharma and clinical trials information on LinkedIn. And they also have a lovely website which you can visit. I'm going to be sharing this recording of this webinar on YouTube, and I will be sharing the link on my LinkedIn page next week. So if you did miss you know, the earlier parts, you can always refer to that. Thank you, everyone, and hopefully we'll meet again soon. Have a good evening. Cheers.
Thank you. All right.
Thank you.
Thank you. Bye.
Thank you so much. Bye.
Thank you. Bye.
Bye. Thank you.
Thank you.
Thank you. Bye.
Thank you. Bye.